My name is Dr. Stephen Kelly. I'm a lecturer in pharmacology at the Medway School of Pharmacy, Universities of Kent and Greenwich. As a university lecturer, my role is threefold. Firstly, in terms of teaching, primarily undergraduate pharmacy students, administration, and that can involve anything from health and safety, admissions, and research, which is could be benchtop research, supervising PhD students, project students. I started off a bachelor's degree in biosciences and then progressed on to an MSc in pharmacology. I then went on to complete a PhD in pharmacology, and then following postdoctoral training, I then progressed on to a university lectureship. My work is almost always focused in terms of pharmacology, which is the study of how drugs interact with the body. However, I've done that through a variety of different types of techniques, from patch clamp electrophysiology, where we look at the behavior of individual ion channels in response to drugs, in vitro experiments where we're looking at the response of a particular organ system in response to drugs, and in terms of biochemistry, we look at changes in protein expression, which we can analyze through techniques such as SDS-Page and Western blotting. SDS-Page, or sodium dodecyl sulfate polyacrylamide gel electrophoresis, is a technique in which we can separate out proteins according to their molecular weight. The proteins migrate through a gel in an electric field, so the proteins that are lighter in weight will migrate faster through the gel than the proteins that are heavier, that have a high molecular weight. We can then transfer the separated proteins onto a nitrocellulose membrane, and then through a process known as immunoblotting, where the antibodies are then conjugated to a dye or an enzyme, we can then quantify the expression of the proteins. SDS-Page is used in many applications, ranging from biochemistry, drug discovery, pharmacology, molecular biology. It's also used in forensics, so it's a very wide-ranging technique. The advantages of SDS-Page over, say for instance, PCR, is that it allows you to look at the expression of the end product, which is the proteins of interest, whereas PCR would look at more of the genetic level in terms of the genetic expression. What SDS-Page allows you to do is look at either the absence or presence of a particular protein, and whether this has either increased or decreased in response to a particular treatment.